All right, guys, happy Friday, week 7. This is fall 2020. We are Bio 208 with Dr. Grenier. So I was just kind of chatting and I needed to make sure I get it on here. Some things that happened in lab yesterday. So in lab yesterday, we recorded about what are the organs, what are the tissues, what are the cells, and how would they be tested in the lab environment. All right. And two things that came up was one of the objectives talks about, again, what are the types of immunity? What are the two types of divisions of immunity? And there's two ways to approach it. Again, big picture is that one type of immunity is the type that's the, I'm sorry, I'm losing track here. That you are human, and because you're human, there are certain types of things that will not come after you because you're a human. And I mentioned like swine flu and avian flu and, you know, deer ticks and things like that. All right. But for pathogens that can use the host as a human and can thrive and live and propagate in the human, you have adaptive immunity to try to prevent them from wreaking havoc to a point where you're dead or dying and they're spreading and moving onward. Okay. And then within adaptive immunity or acquired immunity, what we're going to do is break that into what are your specific defenses and what are your non-specific defenses. Okay. And there are a lot of graphics out there and it gets confusing. All right. That is how I want you to approach it. So in this one here, innate versus acquired, think of innate instead of how it's written here. Think of innate. What am I doing? Think of innate as your, like, because you're human, and think of acquired for you have to have defenses against anything that thinks a human is a good place to go live, grow, propagate, and take over. Okay. And then when we look at our defenses of what happens in acquired immunity, I want you to think acquired is going to be non-specific defenses and then specific defenses. Okay. All right. Now, the other thing that we talked about in lab real quickly after most people had left, but it was a point that I think has been missed, is that we normally don't name any of our lymphatic vessels. However, there is one place that we do give a specific name to the vessels, the capillaries that are the lymphatic system, and that is in the digestive tract. And in the digestive tract, we are going to call the capillaries and the vessels that are lining and pushing into the intestinal villi and are helping to absorb fat molecules. We're going to call those lymphatic vessels lacteals. Okay, now remember, in our blood vessels, I'm just going to try to draw a little blood vessel here. They have all these simple squamous epithelial cells, all right? And let's say this is my blood vessel capillary, okay? And let me get some red, all right? So here's my little red blood cells, all right? And then over here, all right, is, I'm trying to draw a simple columnar, all right? Simple columnar cells that line my intestines, okay? And in between, you know, this is that interstitial fluid, and I am going to have, again, leaking plasma out of my capillaries, and then I'm going to have these, again, specialized capillaries of the lymphatic system that have pores and holes, right? When I am absorbing fat molecules, so let's say I have this fatty acid, okay, and fatty acids are big carbon-rich molecules. When they come into my interstitial system, all right, because they're such a big molecule, it's hard for them to try to go into the blood supply because of the way the endothelial cells are hooked to each other. All right, so instead, part of the way I'm going to absorb fat is fat molecules are going to actually enter my lymphatic vessels, which I'm going to call lacteals in my digestive tract. All right. So one of the things that is important about the lymphatic system in the digestive system is it does actually help with absorption of fatty molecules. And again, fatty molecules are water-hating molecules, so they don't really want to be in your interstitial fluids. They don't want to be in your blood plasma. And so in many cases, they are going to clump together, these fatty, and they're going to form this big kind of ball. If you've ever, how many of you have put oil in a pot before? When you drop oil in a pot of boiling water, the oil kind of makes those little oil balls. Well, that's happening in your digestive tract. And so those oil thickening balls are hard to, again, put into the blood supply because they're not going to stay in solution. So instead, they'll get pulled into the lymphatic capillaries, move through the lymphatic system, and when they are being absorbed that way, we're absorbing them with these lymphatic vessels, which are called lacteals, and they're kind of a big mobby, fatty blob. All right. And eventually we do need to get the fats into our blood supply, but that's going to take place in the liver. So many of these lacteals will, like the blood supply, leaving the digestive tract, they're going to head to the liver. All right. And then in the liver is going to take these fat molecules, and remember, they are going to wrap them with a high density lipoprotein or a low density lipoprotein, and they'll put, like, think of a bunch of asparagus is the fat, and a few strings are the lipoproteins, and they'll basically put little kind of groupings of asparagus and shoot them into the plasma as a smaller, and again with those lipoproteins wrapped around, more water soluble molecule. And then they can move a little bit easier through the system. Okay, so it was kind of missed, I think, yesterday or this previous lectures, and it's in my old lectures, but I just wanted to make sure everybody knows the one place that we name capillaries for the lymphatic system is in the digestive tract. We call them lacteals, and they play a role in, yes, recovery of excess fluid, recovery of the leaking filtration fluid from the plasma, but they also are going to do this other role of helping us absorb fat. So when you go have Popeyes today, and I love me some Popeyes chicken, but it's basically so much fat. All of that fat and your gallbladder is squishing into the lumen, the bile, to help emulsify the fats so the fats can cross through, get into this intercellular area, extracellular interstitial fluid area. Well, again, those fat molecules are still not going to be able to get into your capillaries. So those fat molecules are going to kind of clump together, form this little what's called chylomicron, and they're going to, my friend named her cat chylomicron because she thought that was such a cool word. So I always think of her cat, chylomicron, chylo, and the chylos are going to travel through the lymphatic vessels, those lacteals, towards the liver, and then the liver has a chance to then separate them out, wrap some lipoproteins around them, and then put them back into circulation. All right. So the lymphatic system helps us not just with recovery of fluid here, but with fat absorption from our diet in the digestive tract. So yay, go lymphatics. Okay, so now back to our immune system. Okay, so we are going to talk our non-specific immune system, which is a part of this acquired immune system, okay, or adaptive immune system. And here's the list of 7 key defenses that are part of the non-specific immune system, okay? The first one is that we have physical barriers, all right? So everybody knows that I have a skin. And part of the skin is the sebum, the oil that helps keep the skin kind of pliant. And most of what I see when I look in the mirror is dead, keratin-rich, dead cells. All right. And so the sebum and the keratin helps kind of keep water off and repels water and any pathogens that want to try to come in with any water through my skin. The sebum, like any oil, we don't refrigerate oil, so it's a layer of oil, it's a layer of grease that also kind of prevents things from trying to grow on me. So we don't end up with like coral growing on us or fungus usually growing on us very easily. All right. The physical barriers as we go inside, mucus, spit, saliva, and slime that we create, all of that on top of now the non-keratinized cells and the other simple columnar, simple squamous, and stratified non-keratinized cells, those mucous membranes, those goblet cells, they produce a little bit of slime that, again, tries to keep particles and pollutants and pathogens from easily getting to the cell layer and then maybe pushing through the cell layer. And we see that in the vagina, we see that in the mouth, in the nose. And when pathogens are kind of getting at us, I mean, sometimes that slime, sometimes that goo is a little bit, think about when you blow your nose when you're sick. The slime and the goo is more gunky and it's more full of dead and pus and material that maybe is part of those pathogens that we have kept out, and their secretions. Okay. And then when we look at the stomach, we add, not only do we have simple columnar cells lining the stomach, but we add all that acid. And then think about the urethra and the bladder. The urine is super acidic, usually, most people. It can be basic, but either way, we use pH and those fluids that we accumulate and we secrete into those regions to, again, try to keep anything from trying to live inside those organs and those body waters. And so we have the physical barriers, which are epithelial cells, and then with many physical barriers, we also have the secretions, the mucus, the slime, the oil, the acid, water that also sits and is in those areas, keeping the cells, in some cases, alive and healthy and pliant, and in other cases is being secreted out and potentially what am I trying to say? Not phagocytosed out, but excreted. Okay. So we have some physical barriers. All right. And if I go to my next slide, so again, your epithelial tissue. So all of the linings of any opening, any entry and exit point is potentially your epithelial cells. And again, whatever mucus, slime, wax material that is created could also be part of, again, preventing something from gaining access, preventing from something from just getting a hold onto the skin. And then preventing something from maybe even just living within this region, like in the stomach or in the bladder, very easily, because if they want to live in those areas, they have to live in this very acidic, rich environment. Okay. The next type of prevention or non-specific defense is, let's say, okay, something gets past the epithelial barriers and the mucus and the slime and the oils that were there. Okay. Typically, if you get past those epithelial barriers, right behind the epithelial barrier is areolar connective tissue. And in that lamina propria, that areolar connective tissue, there's some collagen, there's some elastin fibers, there's fibroblasts, fibrocytes, there can be adipocytes, there can be capillaries, but embedded and intermixed in there should be neutrophils, eosinophils, mast cells, and macrophages. All right. And now the eosinophils and the neutrophils, they are capable of phagocytosis, but remember, they're going to release their vessel contents first. Your basophil or your mast cell releases its contents first, and that draws over macrophages and microphages. Your macrophage, remember, its mojo is it eats a pathogen first and worries about digesting and killing it internally. So every cell that does phagocytosis, all right, and or can potentially spit on and break down and help break down pathogens into smaller pieces for phagocytosis, they can live in those connective tissues directly behind the barriers. So if it's like a yard, all right, the fence is the barrier, the epithelial barrier, and we station intermittently many different types of immune cells right behind the fence, so that if something gets past it, they potentially can spit on it and eat it. All right. And because our macrophages are one of our biggest well-researched phagocytic cells, we know that when the macrophage engulfs and eats an entire particle, as this picture up here shows, it is going to merge that vesicle it creates with its lysosome. And its lysosome, remember, and it has some peroxisomes. Those are organelles that are full of deadly digestive enzymes, deadly molecules that can be poking holes into membranes of cells and cell walls. All right. And so when we merge it, that helps break apart our pathogen. And then the next critical piece, that's one of the links to how we get non-specific defenses to help turn on specific defenses, is that our pathogen can be broken into parts, and pieces and parts of that pathogen, and hopefully pieces and parts of the external bits, are taken by the macrophage, taken by the antigen presenting cell, and put on a special protein that is put on the membrane and exposed to the external part of the cell. And that's called antigen presentation. And we're going to learn the protein that's going to hold the antigen from the original pathogen is an MHC complex. And the MHC complex is going to hold our antigen, and it's going to display it on the outside of the cell membrane, so the antigen presenting cell, the macrophage, can go to the T helper cell and say, look, this is what I ate. Here's a piece and part of what I ate. All right. So phagocytosis helps us potentially, second line of defense. If something gets past the barrier, let's spit on it, let's break it apart, and let's eat it. The phagocytic effect of many of our antigen presenting cells, like the macrophage, is also critical that after they eat it, they bring it to the T helper cells. So this is why T helper cells, lymphocytes, will be also in our connective tissues, and you will also be drawn to breaks and tears in the epithelial barriers, and will be drawn when neutrophils and eosinophils and mast cells release vesicles, because T helper cells want to come over so that any antigen presenting cells that eat pieces and parts of whatever got through the barrier can be presented to it, and they can then ramp up defenses if needed to a new pathogen that's tried to gain access. Okay. All right. The next type of nonspecific defense is our natural killer cells. And we talked about this a little bit in lab the last 2 days, how natural killer cells are able to kill cells that they determine are not self, so they're immunocompetent, even though they did not go to the thymus gland. They stayed in the bone marrow, they became immunocompetent. And remember, that's a big thing. We don't understand exactly how they became immunocompetent, but they became immunocompetent. They know the difference between self and not self, all right, just like T cells. All right, so they can go around, and again, think of them as a door-to-door salesman. They can go and be like, all right, I'm gonna go into the ovary, and I'm gonna take the outer quadrant of the very far end of the ovary, and I'm gonna hit every cell, and I'm gonna see who's self and who's not self. Second, I am going to go into the ovary, and I'm going to be, okay, which cells are self but acting and making weird things, meaning they might be hijacked, they might be overrun by a virus. Okay. Third thing, I'm going to go in, I'm going to check and see which cells are self cells that are acting inappropriate, not following the rules of cell cycle, cell regulation, the correct protein synthesis of enzymes, hormones, proteins that are appropriate for that cell and its function, meaning I'm going to look for cancer cells, cells that have turned cancer, turned rogue, and I'm going to kill them. Okay, so that's what the natural killer cells do. And this little graphic here shows you that they are looking for cells, and there's really no cytokine release. There's very little to reason to suspect the cells have something going on. There's not a break, there's not a tear, but they just go through, and they are potentially doing spot checks in neighborhoods of all the cells to see, is there a problem here, and do I need to kill anything? All right. Now, what we have studied is how they kill cells. So if a natural killer cell, and that's what this graphic here is showing you, If a natural killer cell determines, all right, this guy is not acting right, this guy is virus infected, this guy is a cancer, or this guy is not self, we know that they are going to dock onto the cell. They are going to manipulate their intracellular contents with some moving motor proteins to bring their Golgi apparatus and their vesicles that are full of these proteins that are going to penetrate the membrane of my offending cell. They are going to put all of those and line them up so when they release them, it's like a little synapse. The membrane of the natural killer cell is going to release and have new proteins flow out. Those proteins are called perforins, and the proteins are going to cross that little space and start poking holes in the offending cell. Now, anytime I release molecules, even at the neuromuscular junction, even at any synapse of neurons, remember, like, some of the particles and some of the molecules try to go back in the originating cell. So another protein you have to know of the natural killer cells that is present on the membrane to prevent the perforins from turning around and punching a hole in the natural killer cell are known as protectins. Okay, so you have two proteins you have to learn for natural killer cells. Perforins are the proteins that are released with the intent to punch holes. And if I punch holes in the membrane, potassium and proteins and water from the inside of the cell starts to inappropriately leave, and the cell explodes, it lyses, it breaks apart. All right. And to protect myself from that coming back and hitting back on the natural killer cell, I have a second type of protein there that kind of bounces perforin away, which is known as protectin. Okay, so that is how, again, we have this is happening, and the natural killer cells could run into a virus infected cell, a bacterial cell, it can run into an amoeba, it can run into a flute or a worm, anything that could potentially be not self, anything that could potentially be a pathogen that gets in our cells and tries to hijack them, anything, again, cells going rogue, becoming cancerous, it can somehow immunocompetence find those cells and kill them without potentially being drawn over because there was an injury, a break, or a tear in the defenses upstream. Okay. All right. The next two non-specific defenses we're going to talk about are protein molecule-based defenses. So they're not cell-based, they're protein molecule-based defenses. These are interferon and complement. Okay. Now, interferons, and you hear about people take interferon medications if they're on the HIV drug protocols. All right, so again, this is another thing that we really learned about because of HIV virus in the 80s and the 90s. Okay, and this is why Magic Johnson, who contracted the HIV virus back in the day where it was a 10-year sentence to turn into AIDS, he still just has HIV. And he hasn't had full-blown AIDS, and he's not in a hospital bed dying of pneumonia. All right, so interferon is a group of protein molecules. Okay, and interferons could be at times under the blanket of that term cytokine, a molecule released by a cell that can signal immune cells to come over, signal immune cells that help is needed, signal assistance for cell-to-cell communication, that paracrine communication. All right, so what we know about interferons is it's not just one protein, it's a few different proteins. So they'll be like interferon 1, interferon 2, interferon 3, interferon 4, interferon 5, interferon 6. And it's a few different proteins from, again, a grouping of proteins made by any cell of the body. And interferons are usually made by cells that are aware that a virus has infected them and a virus is trying to take over and hijack their system. Okay? And one of the things that interferon is going to do is it is going to be a signal to call over neutrophils, eosinophils, T helper cells, cytotoxic T cells, macrophages, and other immune cells to come over because I am under attack. And chances are I'm calling them over to come over and kill me. Okay? The other thing that some of the interferons are going to do are going to try to turn the cells nearby me that might not yet be infected by the virus. I'm going to try to beef up their antiviral characteristics that would prevent a virus from reverse replication of RNA to DNA or insertion of with clipping proteins inserting their DNA directly into the DNA of the host cell. All right, so that's some antiviral intracellular molecular defenses. Okay, so interferon's going to signal the cells nearby, ramp up these antiviral defenses so you don't get taken over and virus material isn't injected and added to your DNA. All right, it's going to help turn other cells that are already viral infected to, like me, get more assistance from the immune system and also help us start to go through programmed cell death. And if we start to program our death, then we're not as maybe robustly making proteins that are for the virus to be propagating and spreading. Okay, so interferon, again, is like a bunch of cytokines. It's cell signaling molecules, but they are a group of cell signaling molecules that don't have to come from immune cells. They can come from an infected cell to pull immune cells over for help to kill it. They can signal the cells nearby to ramp up your defenses if you aren't taken over yet, and to help ramp up, okay, those of us that are infected, let's go through programmed cell death to slow down and prevent this virus from propagating and continuing to spread. Okay? And again, this is one of the big treatments for why the HIV virus, in some people, they take these drugs and the virus is very, very slow to propagate because we're sending signals to the T helper cells by taking this exogenously through a drug or a pill or a port or IV that you need to ramp up your antiviral. So if HIV virus in our system tries to push in and take over, you can try to push back a little bit and prevent that from happening. Okay. All right. The next big protein group is the complement group. Okay. And these proteins are a different set of proteins that we secrete. And again, they're part of the cytokine grouping that can actually, under the influence of antibodies bound to pathogens, help kill the pathogen. All right. So think of this as a group of proteins that punch holes in pathogens. How do they know what pathogen to pump? How do they know what a pathogen is? Well, they mainly look for where the antibodies are bound. All right. And if an antibody is bound to an antigen, the assumption is the antibody was made to find antigens on pathogens. So we need to punch a hole in this thing because it's a pathogen. All right. And so the method that is the more classical way we understand complement is that if an antibody binds to an antigen and therefore a pathogen, all right, complement is going to come over, and it's the complement C1, C2, C3, and they are going to dock on the part of the antibody that's kind of the flag pointing outward and away from the pathogen. And there's an enzymatic reaction where the protein C3 goes from being turned off to being turned on. And when it's turned on, it calls over some more complement proteins that are going to then punch and make a complex of proteins circling, pushing into the membrane of the cell. That's going to let the cell explode and lyse and break apart. Okay, so again, the most classical way we discovered complement, we named all the proteins in order: complement 1, complement 2, complement 3. We found complement 3 is like the big player that turns on and brings complement 5, 6, 7, 8 to make a complex that punches a hole in the membrane so the pathogen can basically bleed out and explode out and break apart, and then macrophages and neutrophils can come in and eat the rest of it. Okay. And complement is very, very then effective at killing and breaking pathogens into pieces, especially when the antibody is already tied to it. Now, a little bit more research and someone figured out, hey, complement actually can break apart things and the antibodies don't always have to be present. And this is termed the alternate pathway. Now, in this pathway, there's a little bit of how does complement know that it's going to break apart a pathogen cell and not a healthy cell? So some of these proteins that are going to start the process of turning C3 on and making C4, 5, 6, 7, 8 into this complex have to be part of an immunocompetent reaction. I don't understand how it works, but somehow, and it says protein, I can't quite read this, protein D, and B, and properidin, all right? These proteins, they might be released by cytotoxic T cells, they might be released by natural killer cells, they might be released by, again, an unknown entity that I don't exactly know what to share with you, but they are released and they know somehow that the pathogen in front of them is bad and needs to be broken apart. And so they then recruit complement 3 over, complement 3 turns on, puts complement 4, 5, 6, 7, 8 together, punches a hole, and then the pathogen explodes like it would normally. The alternate pathway is a little less dependable, mainly because we're hoping that propridin, factor P, factor B, factor D are there correctly and going to turn complement on to destroy a pathogen cell. But there's no antibody bound, and so we do set up a little bit of room for error. So the alternate pathway is not as robust and not as widely used, but it still is present. And again, anytime I have potential 3 or 4 proteins come together and decide, hey, let's break apart the cell, that could start to happen inappropriately. And that could be part of the reason behind some of these autoimmune conditions. It might not be the reason behind all of them, but it could be the reason behind some of them. And it could be a factor that works with other factors for why in rheumatoid arthritis, your immune system is attacking in and around your joints, or why in ALS and MS, your immune system is attacking pieces and parts in the Schwann cells and the different parts of your neuroglial cells, and then eventually attacking your nervous system, right? It could be why in lupus, again, autoimmune system is happening, okay? What I want you to take away is, again, complement like interferons, they are part of these cytokines. And these cytokines are proteins and other molecules typically made by immune cells or by some of the local cells that are infected to help, again, the immune cells ultimately break apart and kill or beef up defenses against pathogens. Interferon's more about, again, signaling and beefing up defenses and then programming cell death for known infected cells. Complement is how, if, again, cytotoxic T cells and macrophages and neutrophils, there's just not enough phagocytic cells, could be another way to help break up pathogens so phagocytic cells can come in and eat up the pieces. Okay? All right. So, last 2 parts of nonspecific defenses are inflammation and fever, and they kind of go hand in hand. All right? So inflammation is defined as some type of response to injury, and typically inflammation can be localized or it can be whole body. Okay? So when we normally get a cut or we get a break in the epithelial tissue, cytokines are being released by neutrophils, mast cells, eosinophils. All right? We have potassium, we have adenosine, we have guanine, we have RNA, we have amino acids, we have all these molecules that are supposed to be inside the cell, ADP, GDP, we have phosphates. They are now part of the extracellular environment. So just by pure Osmosis rules. Number of solutes in an area of an injury have increased. So water wants to come to it. So the swelling, the edema, the accumulation of water in an injury site is just following normal osmosis rules. Second, when we usually have an injury, again, mast cells, neutrophils, and then the pullover of all of a sudden endothelial cells are being exposed to more high levels of potassium, adenosine, ADP, phosphates, things that are supposed to be inside the cell, just like we talked about in the clotting process. They are going to signal there's an injury, I'm an intact vessel, I'm going to dilate so more blood flow can come into this area in my secure capillary and vessel, and I can get potentially more neutrophils, more lymphocytes to this area. As a consequence of dilation, remember that's going to bring more fluid in. Okay, when I bring more blood flow to an area, I'm bringing more red blood cells, I'm bringing more heat. So heat, swelling, redness is somewhat because of the areas and capillaries and blood vessels not damaged, but nearby are dilating, bringing more blood, more energy, more neutrophils, more lymphocytes to the area. The vessels or anything that's injured, remember, are going to constrict. And they're going to constrict to minimize potentially red blood cell loss and fluid loss to the extracellular environment. Okay, so that's another reason why the swelling. That's some reasoning behind the heat, because remember, blood travels at a little higher temperature than the extracellular areas, and why the redness occurs. Now, if I have a lot more phagocytosis, a lot more ATP being utilized for spitting out vesicles, bringing phagocytosis contents in, more ATP expended, that's also a little bit of a reasoning behind the heat. All right, so inflammation is usually swelling, redness, heat, and then again, because the nerves can be more sensitive because they're being interacting with things that are supposed to be inside cells, and they're no longer inside cells because cells have exploded. So you have more pain, more sensitivity, more squeeze from the swelling. That's that tenderness, right? And the bigger the injury, the bigger the area that's probably going to be swollen, red, hot, and tender. And if it's big enough or this happens long enough, that inflammation can even become more systemic. Now remember, some of the cytokines we're releasing are going to change blood flow. Some are going to call in more neutrophils, more eosinophils, more macrophages, more lymphocytes. Some are going to head to the bone marrow, because if I have an injury site and I'm about to use a bunch of my white blood cells, I need my bone marrow to start ramping up the next grouping of lymphocytes and white blood cell replacements for the cells I'm about to use and lose. Some cytokines are also going to head to my brain. And a group of these cytokines that head to my brain can go to the hypothalamus and go to the set point area and try to raise my whole body temperature from about 37 degrees Celsius to 38 degrees Celsius, which is like 98 degrees to about 100 degrees. Okay? And part of the reason why we raise the temperature, and you should see this in your chemistry class, this is another reason why you have to take chemistry, is if I take water and I make it a little bit warmer, then again, from a level of molecules are in constant motion, in warmer temperatures, molecules are going to move at a higher rate. If I raise the temperature, some of my chemical reactions can now happen with molecules in constant motion and motion happening at a faster rate. Some of these chemical reactions that I need to help kill, to help destroy, to help clean up, they are going to happen at a higher rate. So part of raising my temperature and getting my, again, body water to warm up is to get chemical reactions and metabolism, the rate of breaking and making molecules, to go at a higher level. Okay? The next part is many pathogens live in water and they want the water to be a certain temperature. So the second reason to raise my temperature is it might kill some of the pathogens just because they want to live in 37 degrees Celsius water and not 38 degrees Celsius water. So the fever is going to typically result when I need my immune system to ramp up the chemical reactions, to ramp up the activity, and to maybe just make the body waters a little bit warmer to try to prevent some of the pathogens from surviving in that environment. So fever is supposed to be a good thing. And I remember this old, I think it's before even Grey's Anatomy. It was the one with George Clooney back in the day, where they were all working in a hospital. And the guy from Revenge of the Nerds and Top Gun, he was Goose. I forget his name, but he's always Goose to me. All right, they were all doctors. ER, that was the show. And I remember one of the times they had a patient, and she was a little girl, and the doctor was from some foreign country. And they were like, can we give her some Tylenol for the fever and the pain? And he was like, no, fever is good. That's the body fighting the infection. And they were like, well, in this country, we don't want to put people in pain if we don't have to. And he was like, well, then it's a little bit of a toss up, because you want a little bit of fever because it helps your body fight the infection, but it's a little painful because remember, temperature receptors turn into pain receptors when they get a little too hot. And so that's part of the reason why temperature hurts, especially temperature of our system. In pain will be pain, feel like pain in our joints, pain in our areas. And so a little bit of heat and a little bit of pain, if you can tolerate it, are actually helping you fight the illness, fight the pathogen. It's a slippery slope. As we continue to fight the pathogen and continue to release more and more of these cytokines and more head to the brain and more tell the brain to raise the temperature, we can start to raise the temperature to a level where, remember, molecules inside us are still made of ionic and covalent bonds. And if it's too hot, the atoms, the electrons can move too much, and those molecules can break apart, denature. And this is how, like, they back in the old school days, they crack an egg and like, this is your brain. And then they crack the egg over the hot pan. This is your brain on drugs, and it's cooking. All right. Well, that's not really drugs, but that's just high temperature. All right. What you're seeing when you cook eggs is you're seeing that some of the proteins and some of the molecules under all that extreme heat are denaturing, and they're changing from being maybe swirly twirly and this molecule that works a certain way, and they're becoming no longer a workable protein, no longer a workable enzyme. And that's the big issue of why we don't want fevers to get too high, and we don't want fevers to go on too long, is because then cells that have functional and necessary proteins and enzymes start to see their proteins and enzymes denature and die and no longer work. And then we're not even fighting the pathogen anymore. We're trying to keep our own cells alive. Okay? So a little fever is okay. Too much fever is bad. And then when you think about how big we are, remember that little kids, if they're 70% water and they weigh 40 pounds, and 70% of that is water, that's 30 pounds, let's say, just because I'm making math easy. And if you lose 2 pounds of water from sweat with fever, you are dehydrated at a dangerous level very quickly. All right. And so that's part of the reason why when and if you ever have children, your threshold to be worried about fever in a baby is 100.4. Your threshold for being worried about fever in an adult is a 104. And part of that goes to when I have fever, I'm going to start sweating. I'm going to start losing water. All right. And I'm probably not going to feel like drinking, and I'm probably going to be sleeping, and I'm probably going to be in a lot of pain, and I'm not going to be taking fluids in very much. So the fluid I'm losing is faster than the fluid I'm gaining, and I start to get dehydrated. And that dehydration ratio of being in a dangerous zone happens faster in smaller bodies than it does in bigger bodies. And so that's why another reason why we start to worry about fevers in little children that spike up and happen really fast and really long for, again, 100.4 versus 104, right? All right, so those are our non-specific defenses. And then again, here is a summary slide. I'm going to stop the recording here so I can yell at my children.